A genome is the complete set of genetic material, DNA or RNA, present in an organism. It contains all the information needed for the growth, development, functioning, and reproduction of that organism. Genomes are found in the nucleus of eukaryotic cells and in the cytoplasm of prokaryotic cells. Key features of genomes and how they contain genetic information. 1. Genetic material. The genetic material in a genome carries the instructions necessary for building and maintaining an organism. In most living organisms, the genetic material is composed of DNA, deoxyribonucleic acid, although some viruses have RNA, ribonucleic acid, as their genetic material. 2. Genes. Genes are segments of the genome that encode specific instructions for the synthesis of proteins or functional RNA molecules. Each gene carries the information needed for the production of a particular protein or the regulation of a specific biological process. 3. DNA structure. DNA is a double-stranded molecule made up of nucleotide units. Each nucleotide consists of a sugar, deoxyribose, a phosphate group, and one of 4 nitrogenous bases, adenine, A, thymine, T, cytosine, C, and guanine, G. The sequence of these bases along the DNA strand forms the genetic code. 4. Base pairing. The structure of DNA allows for specific base pairing. Adenine, A, always pairs with thymine, T, and cytosine, C, always pairs with guanine, G. This complementary base pairing is crucial for the accurate replication of DNA during cell division and the transcription of genetic information into RNA. 5. Coding and non-coding regions. Genomes have coding regions, which contain genes that encode proteins or functional RNAs, and non-coding regions, which do not code for proteins but may have regulatory functions. Non-coding regions can include promoters, enhancers, and other regulatory elements that control gene expression. 6. Chromosomes. In eukaryotic organisms, the genome is organized into chromosomes within the cell nucleus. Chromosomes are structures composed of DNA and proteins. Humans, for example, have 23 pairs of chromosomes. Prokaryotic organisms, such as bacteria, typically have a single circular chromosome in the cytoplasm. 7. Genome size. The size of a genome varies widely among different species. Some organisms have relatively small genomes, while others have large and complex genomes. Genome size is not necessarily correlated with organism complexity. For example, some plants have larger genomes than humans. 8. Genome sequencing. Genome sequencing is the process of determining the complete DNA sequence of an organism's genome. This technology has advanced significantly, allowing researchers to decipher the genetic code of various organisms. The Human Genome Project, completed in 2003, was a major international effort to sequence the entire human genome. 9. Genetic variation. Genetic information in a population is subject to variation. Genetic variation arises from mutations, genetic recombination, and other processes. It contributes to the diversity observed among individuals within a species. 10. Evolutionary significance. Genomes play a central role in the process of evolution. Changes in the genetic material over time, driven by mechanisms such as natural selection, mutation, and genetic drift, contribute to the adaptation and diversification of species. 11. Epigenetic information. In addition to the DNA sequence, genomes also carry epigenetic information. Epigenetic modifications, such as DNA methylation and histone modifications, can influence gene expression and play a role in regulating cellular processes. Understanding the structure and content of genomes is fundamental to advancing our knowledge of genetics, genomics, and the molecular basis of life. The information encoded in genomes governs the inheritance of traits, the development of organisms, and their interactions with the environment. Advances in genomics continue to have profound implications for medicine, agriculture, and various scientific disciplines.